Sensory system is one of the four controlling systems. Sensory, nervous, endocrine, and circadian systems are controlling and regulating all the other organ systems. Sensory system is a part of nervous system. It is an outgrowth of it. It is a special modification of the nerve cells. Sensory receptors are modified neurons. So, just like all other neurons, they use synaptic transmission to impart information to the next neuron, and next one, and next one, and next one. All senses work along the same principles. So, there's going to be a stimulus that comes either from the external environment or the internal environment. It is detected by a sensory receptor, which is a specialized type of cell. And then inside the cell, there's going to be a chemical cascade. That cascade is called transduction. So whatever stimulus comes to the outside of that cell is going to trigger changes on the inside of the cell. And those can be changes in the membrane potential, they can be enzymatic changes. They're going to change the properties of the cell. That is called transduction, this change in the properties of the cell. So it's going to end up with, let's say, an action potential, which is going to uh, result in synaptic transmission of that information that the sensory cell has been triggered, and that information is then going to be transmitted to the next nerve cell, to the next one, to next one, next one, until it gets to into the appropriate area of the central nervous system, which is usually the brain, sometimes the spinal cord. So you start with a stimulus, then whatever is happening in the inside of the cell is transduction. Then there is a transmission from the initial sensory cell to the rest of the nervous system. And then the central nervous system is going to interpret. So there's interpretation, that information. So that interpretation means integration and processing over time. It can be processing of just that one stimulus, and it's also integration of uh, all the stimuli coming from all the sensory systems together that might uh, put together a broader, more complete picture of what is happening either in the internal or in the external environment. Then the central nervous system is going to make a decision and is going to trigger an action, necessary action. That can be behavioral, like movement. It can be physiological, secreting hormones or whatever for the maintenance of homeostasis. Uh, so nerves that bring that sensory information from the periphery, from the sensory cells to the central nervous system, to the brain, those are called ascending pathways. And you remember the descending pathways are the motor pathways, the pathways that go from the central nervous to the periphery to tell the periphery what to do, to the heart to speed up, for the kidney to slow down or speed up filtering of the urine, or to the muscles to move. Um, so ascending pathways are those going into the center from the periphery. Those pathways are going to end in particular parts of the brain, mostly in the thalamus. From various different senses, they end up in the thalamus. There are some exceptions. Some parts of somatosensation and olfaction, that smell, don't go to the thalamus. They go to hypothalamus, among other places. Uh, then from the thalamus or hypothalamus, these nerves are now not going called nerves anymore. They are now tracts because they are in the central nervous system. They go to higher level processing areas, and those are primary sensory cortices. Uh, and for instance, for vision, the visual cortex is in the occipital part of the brain, in the back of the brain. For hearing, it's the temporal parts of the brain. Somatosensory is, remember, on the sides of the brain here, on the kind of on the top. Olfactory cortex is right above the nose in the brain. So there are several different places where different senses are processed. Those are primary sensory cortices. And then information from the primary sensory cortices goes to kind of higher level association areas and the multimodal integration areas where information is put together. It comes from multiple senses. Like, from the stress response, like if you're a zebra, you smelled the lion, you heard a lion, you saw the lion, all of that gets put together. So we have uh, a lot of different senses. You heard five senses. We have many, many more than five senses. There are general senses and special senses. General senses are generally called somatosensation. And then special senses are vision, olfaction, that's smell, gustation, that's taste, audition, that's hearing, and equilibrium, that's balance. Other animals uh, pretty much all have the same sets of senses, but sometimes might perceive it slightly differently. They might be, let's say, on a different part of the spectrum or process them a little bit differently. Uh, and there are some animals that are capable of detecting other aspects of the environment that we cannot. For instance, the electrical fields and the magnetic fields, pheromones that we cannot. So let's start with the general senses. So somatosensation is a collective term for a whole bunch of general senses that are distributed throughout the body. Um, so some of those senses are exteroreceptors, meaning sensing the information from the external environment. Some of them are interoreceptors, which means they are detecting the information from the inside of our environment. So, for instance, for the outside environment, a lot of those senses are in the skin, mostly in the dermis, where we have the senses of touch, light touch, pressure, which means it has to last a little longer, tickle and itch, pain, vibration, hot and cold, hair movement, those kinds of things. Are extra receptors that are all in the skin, which includes specialized sensory organs like Pacini's, corpuscles, Meissner's corpuscles, Merkel's discs, and Ruffini's corpuscles. They're all in the skin, but some are not specialized. Some are naked nerve endings in the skin that sense pain and temperature. Then we have those that are internal. They might sense stretch, like stretch receptors in the muscles are going to tell us are they stretched or not, or contracted. Stretch receptors in the uterus, stretch receptors in the nipple. Remember we talked about them when we talked about the positive feedback loops. There are pain receptors in the inside. Proprioception is kind of a general term for all the sensory cells that are telling us about the position of our body in space. Kinesthesia is kind of a generalized term for all the sensory receptors that tell us about our movement through space. So, a lot of stretch receptors in various muscles and tendons, touch receptors in various places. Uh, so some of those senses are mechanoreceptors. Most of those senses are mechanoreceptors, but there are also some that are chemoreceptors. They are sensing chemicals. For instance, some of the naked nerve cell endings for pain are not mechanoreceptors. It's not that you kind of move the nerve cell and it feels it as pain, but pain is coming from the damage of the cells neighboring to it, and those are going to release chemicals like a cell breaks up and cytoplasm comes out into the intercellular space. There are molecules in it, and these nerve endings can sense those chemicals. Uh, there are also chemoreceptors inside of our body, mostly on the inside walls of our arteries and veins and heart, and they measure concentrations of oxygen, concentration of carbon dioxide, concentration of glucose. Uh, there are also mechanoreceptors that measure blood pressure inside blood vessels, inside the kidney. All of those are somatoreceptors. Uh, Trigeminal nerve, or the cranial nerve number five, is uh, is interesting because it is um, innervating a lot in our head, including the inside of our mouth. And it has membrane receptors at the ends of the nerve endings. And those membrane receptors can bind the molecule capsaicin. Capsaicin is the molecule that makes hot peppers hot and all the other spicy foods hot. So if you're eating hot food, it's because you are capable of sensing the molecule capsaicin by the endings of the trigeminal nerve inside of the mouth. So this is a specialized chemoreceptor which also serves as a pain receptor. It has evolved to get us like not to eat hot stuff. Now we have culturally learned to like hot stuff. Some of us, at least, like spicy food. But in general, hot peppers are hot in order to prevent us from eating them. Why? Because if you eat a hot pepper, which is full of vitamin C and it's good stuff, juicy stuff. When we swallow it, the seeds that come through our GI tract and exit at the other end are not going to be viable. There's not going to be any hot peppers coming out. So that's what happens when a hot pepper seed goes through a mammalian GI tract. On the other hand, birds don't have capsaicin receptors. They see red pepper, and they're specially attuned to things that are red, and they'll go and eat it and get a wonderful meal full of vitamin C, and they swallow it, and they swallow the seeds. Yet the seeds that go through the bird GI tract, when they come out on the other side, they're perfectly viable. They're going to grow into another hot pepper plant. Birds are going to disperse far away because they fly. And when are they going to poop? Usually when they're standing on a branch or on a tree or on a bush or something like that. And it's going to poop under. And hot peppers like growing in the shade. So it's kind of a perfect organization for the hot peppers to propagate themselves. Uh, There are several different somatosensory pathways because there's so many different sensory cells, different mechanoreceptors and chemoreceptors inside of the body, outside on the in the skin, measuring things that are changing in the inside environment and the outside environment. So of course there are all sorts of different pathways to the brain from all these different systems. Uh, so they go from the spinal cord. Uh, they go into the spinal cord via dorsal root ganglia, follow the spinal cord up into the brain. Inputs from the left side of the body cross over to the right of the spinal cord and are processed by the right side of the brain. And inputs from the right side of the body cross over here, go to the left side of the brain for the processing. Everything that's happening above uh, kind of throat is going to be processed on the same side. Everything that comes from under the throat is going to cross over and is going to be processed by the other side of the brain. Uh, Somatosensory cortex, that means all those touches and pains and all that stuff, is organized like a map of a human body here. That's what is called a homunculus, or a little person, a little man. Uh, and so you're going to have parts of the body that have a lot of sensory inputs, like the fingers and the lips and the tongue are going to be represented. That homunculus, that little human, is going to have very large fingers and lips, while where we have fewer sensory receptors, like on the skin of our back, that's going to be smaller. But anyway, it's kind of organized like a human. And right next to it, on the other side of the sulcus, is the motor cortex, which maps exactly the same way, like a little homunculus. So wherever there's a little, part of the somatosensory cortex that corresponds, let's say, to the left thumb, there's going to be right across the sulcus, there's going to be a center for motor control of that right thumb. Well, left thumb would be here. So it's going to tell muscles of the thumb to contract. So there's a representation of the body in the somatosensory cortex, which maps closely to the motor cortex right next to it. Okay, and then we get to the special senses. All right, let's start first with hearing or audition. Audition is a mechanoreceptor. The ear is a mechanoreceptor because it is uh, measuring the mechanics of mechanical pressure of the air movement. So the sound is going to produce oscillations of the atoms and molecules that make up the air. And those oscillations are going to go through our auricle. It's called an auricle in humans because we cannot move our ears with our muscles. In animals that can move their ears around, this part is called the pinna. And so those oscillations get captured there, kind of narrowed down. This is a funnel that works to bring those oscillations into the auditory canal, at which end is the tympanic membrane or eardrum. So that tympanic membrane is going to start oscillating at the same frequency as the air. The air molecules are going to beat on the eardrum and it's going to force it, mechanically force it to oscillate at the same frequency. What is another word for frequency? Pitch, right? So if you're hearing music, different notes are going to have different pitch, meaning different frequency, and are going to force the tympanic membranes of both ears to oscillate at exactly the same frequency. Uh, the This is where we end with the external ear, and we are now getting into the middle ear. The tympanic membrane that is oscillating is attached on the inside. So this is, the sound has come from the outside into the left ear, and this is the left eardrum, left tympanic membrane. It's oscillating, and attached to it are the ossicles of the middle ear. Those are tiny little bones. Those are the three and three on the left, three on the right, six smallest bones in the human body. And they come in this order. Malleus comes first, hammer, and then attached to it is incus or anvil, and attached to it is stapes or stirrup. And so when the air is forcing the tympanic membrane to oscillate at a particular frequency, the tympanic membrane forces the hammer, which forces the anvil, which forces the stirrup, to uh, oscillate with that same frequency. So they're transmitting the vibration from the outside air through the membrane, through the three bones. The last one, the stapes, is attached to the cochlea. That is now inner ear. And it is attached to the cochlea at a spot that is called the oval window. Uh, in order for all of this to happen well in the middle ear, the pressure in the middle ear has to be equal with the pressure of the air on the outside. Because if it's higher than the outside, then the movements of the air molecules is not going to be able to push the tympanic membrane to oscillate at a high enough strength or at all, or if it's too low. So either way, the inside and the outside of the tympanic membrane has to be the same air pressure. How do we get the air pressure to be equalized? Through what is called eustachian tube. The middle ear is connected to the throat, the pharynx, by eustachian tube. That's why, let's say, when your ears are hurting on an airplane when you're landing, you try to push the air up and you feel the air opening eustachian tubes, and which equalizes the pressure in the middle ear, and so your ears don't hurt anymore. Uh, Eustachian tube is usually closed, but it opens when you're doing that. It opens during yawning, it opens during swallowing, and allows the air pressure to be on exactly the same in the inside of the middle ear as it is when the outside air. So the now we are in the inner ear. Remember, the stapes is transmitting the vibrations to the cochlea. What is a cochlea? Cochlea means a snail because it's a spiral. It's a spiral tube. Spiral tube. And it's a long tube made of various membranes. It's outside membrane, a couple membranes on the inside that are dividing the inside into several different chambers. This tube is filled with liquid. Uh, so there are chambers, like as I said, if you look at the cochlea at a cross section, it is going to be divided into chambers. So there's the snail goes around. The top of the snail is scala vestibula. The bottom of the snail is scala tympani, and then in between them, the middle uh, segment is the scala media, and the scala media is what all the interesting stuff happens. Uh, in Inside of scala media, there is another membrane called the Reissner's membrane, and it divides the cochlea, the liquid in the cochlea, uh, between the scala vestibula and scala media. The basilar membrane separates the scala media and scala tympani. Organ of Corti, or spiral organ, sits on the basilar membrane. That's inside of scala media. And organ of Corti is the business end of the ear. Organ of Corti is the sensory organ within the sensory organ. This is what actually perceives uh, the sensation of uh, of hearing. And because it is made, organ of Corti is made of sensory cells. The highly modified neurons, those are mechanoreceptor cells, that are called hair cells. And they're hair cells because on the surface they have cilia. They are called stereocilia. So when you hear something, there's all this mechanical vibration uh, that is going to be transferred to the cochlea, and the fluid inside of the cochlea starts to oscillate. It moves the basilar membrane. And that basilar membrane is going to move. It's going to move organ according with it, and there's a pictorial membrane on top of it. Uh, that the that is uh, the Reissner's membrane. So the pictorial membrane is covering the stereocilia, right? It's sitting on top of those hair cells, and as it moves, because the fluid is moving, because of all the mechanical agitation, uh, that membrane is going to move the hairs. And when it moves the hairs, those hairs on the top of the membrane of the hair cell, when they move in a particular direction, they're all going to uncover a hole that is an ion channel that allows positive ions, mostly calcium, some sodium, to go into the cell, which is going to change the polarity of the cell. It's going to depolarize the cell. It's going to stimulate that cell. If it's moving the opposite direction, it's actually going to close it better. There's always a little bit of opening. There's always a little bit. It's kind of a leaky channel. It's a leaky ion channel, so there's always a little bit of ions going in and out. If you open it more, it's going to surge in. And if you close it to the other side, it's actually going to completely close any kind of leaking of ions. Uh, if the pitch of the sound is very high, the high frequency, only the very first portion of the cochlea vibrates. Remember, the stapes is attached to the oval window, which is the beginning of that snail, of that spiral. So, if it's a very high pitch, only the first part is going to vibrate, and only the hair cells at that part are going to vibrate. Everything else is going to stay quiet. On the other hand, if it's a very low frequency, low pitch, deep bass sound, then the first parts are going to stay still, and the end of the cochlea is going to be agitated, and the pictorial membrane is going to move on top of those hair cells, moving the stereocilias away, and thus opening the channels, making them depolarize, making them stimulated. So organ of Corti, and one on each side, is actually going to detect the pitch by which part of of the organ of Corti, which part of cochlea is vibrating. That gives you the pitch. Even better, when a particular pitch, you're hearing a particular note. Uh, so let's say you're hearing A, you're going to have the stimulation of the hair cells in the organ of Corti that are attuned, where the membrane is attuned to A. It's at the same time, so it's going to move those hair cells, the stereocilia, in such a way that it completely opens the channels. At the same time, the neighboring cells, the A sharp and A minor, A flat, A sharp and A flat, those are actually going to be inhibited. The hair cells are going to go in the opposite direction, completely closing any kind of influx of ions, making for more of a contrast, making for a clear hearing of that tone and not something fuzzy. Okay, so uh, the cells that are triggered, that are opening ion channels, so potassium and calcium can surge into the cells. That triggers a membrane potential change, which is called the receptor potential, uh, and that opens sodium channels, specialized sodium channels, which release a neurotransmitter, which induces an action potential in the next cell. And next cell is auditory neuron, and the auditory neuron is going to go to the temporal areas of the brain where sound is processed. How is that part of the brain going to know what tone did you just hear? It's going to know because it'll know exactly which neuron came from which part of the cochlea. So if you hit an A, then there's going to be a particular axon going to it. So if the brain gets the information that that particular axon is firing, that means it was A, and it was not A sharp or A flat. Um, so that's how we hear. Um, so when you think about it, the animals that have uh, cochleas of about our size, they're going to be able to hear the range of pitches just like us. Much, much smaller animals that have tiny, tiny little cochleas, they can hear only high pitch. And because those cochleas are not just shorter but also thinner and smaller, they can hear even higher pitch than us, right? That is ultrasound. We cannot hear it, but mice and a lot of other small animals, voles and whatnot, can shrews, they can hear ultrasound. On the other hand, very large animals are going to have very large, long, thick cochleas, and the tips of those cochleas can perceive frequencies lower than we can. Um, it's just longer. We don't get to go that far. And so a lot of large animals, elephants, whales, rhinos, giraffes, tigers, they can perceive infrasound, and often they use infrasound for communication with each other, especially because infrasound carries very long distances through the substrate, like through the soil, and not while higher frequencies go better through the air. So infrasound and ultrasound, that just refers to our limitations. Other animals can hear infrasound or ultrasound just the same way with the same mechanism as we hear our range. Okay, um, so we are hearing the sound through two ears, right? The sound comes, the air vibrations coming to left and right ear, and we have hair cells responding, sending information to the temporal side, temporal primary sensory cortex for the auditory information on the same side, and the temporal lobe. Uh, Before it gets to the temporal lobe, it goes through some other nuclei uh, in the brain. Remember, everything goes through the thalamus, and thalamus is going to have a bunch of nuclei. So the auditory information is going to go through something called an olivary nucleus. And the olivary nucleus receives information from both ears. So there's information coming from the left, information coming from the right, and they meet in the olivary nucleus. Olivary nucleus can then compare what the left ear is hearing to what the right ear is hearing. And that helps us figure out where the sound is coming from. Because let's say the sound is coming from my right. My right is going to hear it with higher intensity. So the vibration in the tectorial membrane is going to be stronger. So it's going to send a stronger stimulus through the olfactory nerve than this one. Then also it's going to get that sound earlier than this one. There's going to be a difference in milliseconds or microseconds between when this ear hears it and this ear hears it. Olivary nucleus can sense this and can measure the intraoral intensity difference and intraoral time difference. So how intense the two ears hear it? Is there a difference? And what is the timing difference? When does the sound come to the left and the right ear? And from there, calculate exactly where the sound came from. Uh, so that is happening in the olivary nucleus, and then from there, auditory processing moves to a nucleus called inferior colliculus, and thalamus, superior colliculus, uh, medial geniculate nucleus. You don't need to know all these various nuclei. It ends up in the temporal lobe where the auditory uh, cortex resides. Uh, on the way there, it's already going to compare itself to some information that comes from the visual system, from somatosensory system. So even when it by the time it gets to the temporal area, there was already some comparison between the information coming from different systems. Uh, Different parts of the temporal lobe then process different types of auditory information, like music and speech and things like that. There is Wernicke's area that processes auditory and visual information that is about understanding language, spoken language and written language. If that is damaged, you have trouble with speech because you don't understand the language. You can't read, you can't understand what people are talking to you. Remember, a little bit to the front of it, there is a motor area called Broca's area, which, if it's damaged, also have a speech impediment or you can't speak because that motor area is stimulating all the muscles of the lips and tongue and throat that are involved in the mechanics of speech. Okay, so while we are in the middle ear and an inner ear, we are going to talk to the next one, which is balance or equilibrium. It's very similar to hearing. I mean, there's cochlea, and then it's kind of attached to the cochlea is the sense of balance. It is also a mechanoreceptor. It is also hair cells. It is also overlying membranes that move the stereocilia. For balance, they're now not called stereocilia, they're called kinocilia. Also open ion channels when those cilia are displaced by a membrane. Also in the inner ear. So they're related. The hearing part of the inner ear and the balance part of the inner ear are very similar the way they work, and they're found in the same place. Uh, so sense of balance, that means the sense of head position, head movement, and body movement. So head position is sensed by the organs in the inner ear called utricle and saccule, while the head movement is sensed in the semicircular canals. The utricle and saccular are the sacs. They are on the inside. They have a macula, which is a tissue that is made of hair cells, and those kind of cilia are covered by what is called an otholitic membrane. It's a doesn't really look so much like a thin membrane, more like a layer of a gel, which acts as a membrane because it's suspended in liquid, and so the gel is going to act like a membrane, and occasionally it's going to touch and displace the hair cells if you change your position. On top of that gel, on top of that otholitic membranes, there are crystals. They're calcium carbonate crystals, and they're called otoliths or otoconia, and they're on top of the membrane. So they're kind of pushing the membrane down, making it top heavy, and so that it's pushing down onto the hair cell. So when the head moves, the otholitic membrane moves in the opposite direction. The head moves to the left, and the membrane stays back, and that moves the cilia in that direction, opening ion channels, triggering the signal. Uh, so different hair cells uh, depolarize while other depolarize because of the movement, right? When you have that membrane moving around them, you have one in the left ear, one in the other ear. They have the utricles and saccules in different orientations, and so the macula is in different orientations. So whenever you make a movement, some of those cells are going to be stimulated and others are going to be inhibited, some depolarized, some hyperpolarized, and this is the information that is encoded as the direction of the movement that then the central nervous the brain can interpret in its primary equilibrium cortex. Uh, and then we have three semicircular canals. They are circular tubes. One is horizontal, one is vertical at 45 degrees, and the other one is vertical but is 90 degrees turned. So, uh, so horizontal, vertical 45 in, out, vertical 45 in. So then there are two inner ears, so there are six semicircular canals detecting six different orientations. And the base, the very beginning of each canal, is a little wider. It's called an ampulla. Ampulla has hair cells. They're gathered in a structure. It's called a crista. Membrane here is called cupula. It sits on top of the cilia. As the head moves, the liquid inside of the canal lags behind the motion. It's slower than the motion, and it moves the membrane, the cupula, over the hair cells, stimulating them or inhibiting them. Exactly the same principle as hearing, as the utricles and saccules. And then the brain takes all that information from all the sacs and all the six canals and interprets the position or the motion, the directional motion, speed of motion of the head. And that's taken by vestibulocochlear nerve. From there to the brainstem and to the cerebellum. Cerebellum is important for balance, as well as a center for equilibrium in the temporal parts of the brain. Okay, um, so there's more processing and tracts and whatnot. There's inputs from some of the uh, other cranial nerves that are using visual information and other information to put it all together. So we have a better sense of our position in space and balance. Uh, and Interaction with the visual system is also important in that we get what is called the vestibulo-ocular reflex. So when your head is moving, you can still keep your eyes in one spot, right? Looking at one thing. Compensates for movement, stabilizes the image of the retina while you're moving your head. So they are important to work together. All right. Now we're going to go to photoreception, vision. Uh, so that is a very, very specialized sensory organ. The eye is a very specialized organ because it's sensing light in the environment. What is light? The photons, right? That's radiation. Photons have energy, and that energy is then used to energize molecules that have color. So if a molecule is colored, has color in it, it's called a pigment, and that kind of molecule can be energized by a photon. So when we talk about photoreception, that's kind of a broader sense of being able to pick up photons and being able to sense them and respond to them in some way. Vision is a little bit narrower. It refers specifically to the sense to the part of photoreception that gathers photons in the environment in such a way that it composes an image of the outside world. So we can see the objects, we can see their edges, their color, their movement, uh, and forms that image in our heads. Uh, that is not all of photoreception. Not all photons that we detect are used for forming the image. There are also there's also such a thing as non-visual photoreception that is used by the body to control some other things. One is mood. If you are sitting in constant darkness and you never see the light because you are living in a cave, that is going to make you depressed. If you live in a prison where they never turn off the lights, you're going to get depressed. So it's going to affect the mood. Pupillary reflex is the narrowing of pupils in intense light. It doesn't have to do anything with the images that we see. It needs to be a light meter. It needs to measure how many photons per unit of time are coming in, and in order to tell the pupils to close or open in order for to regulate how much light is coming into the eye. Entrainment of the circadian rhythms. So the circadian clock needs to know how bright it is out there. Again, it doesn't care about the images, it cares about the intensity of light and the wavelengths of light out in the environment. So those all those functions, the mood, the pupillary reflex, the circadian rhythms, those are controlled uh, by a light meter, and light meter is found in a retina, and it's only about 1,000 cells in each eye, in each retina. Those are ganglion cells, not bipolar, horizontal, or rods of cones or anything like that. Those are ganglion cells. Those are nerve cells proper, and they about 1,000 cells in each eye, and they are using a photopigment called melanopsin. Melanopsin is the pigment that is energized by photons and provides information about the intensity and wavelength of light, which is important for mood, pupillary reflex, and entrainment of circadian rhythms. Uh, Vertebrates that are not mammals, like birds and amphibians and reptiles and fish, uh, they have photopigments in other parts of the brain. For instance, the pineal is directly responsive to light. There are parts of the hypothalamus that are directly responsive to light. Light can penetrate through the skull and the brain and trigger the stimulation of those cells that have those uh, pigments, a whole bunch of different pigments like pinopsin and pineal. There are some animals that even have a peripineal organ. So there's a pineal organ gland and a peripineal right next to it, and it even has a lens that is on the top of the skull, which is designated not for formation of images but for detecting the light intensity, like in lampreys and tuataras. Uh, so we're mammals, so we don't have any of that exciting stuff. All of our non-visual photoreceptor is happening in those 2000 cells, retinal ganglion cells that are expressing the photopigment melanopsin. Uh, Vision or photoreception is sensing radiation in a particular segment of the radiation spectrum, which we call visible spectrum because we can see it. Of course, there are going to be animals that can see different spectrums than us. Some of the animals don't see red very well. Uh, some can see into the ultraviolet, some can see into the infrared. Uh, so, infrared, remember, is also hot. So, detection of infrared light is photoreception, but it's also thermoreception because it's detecting heat. So, some snakes can detect infrared because they want to detect the body heat of a rodent or whatever prey they're going to try to catch. There are beetles that have sensors of infrared receptors, and they're using it to detect the light of a burning forest, often tens of miles away. Those are beetles that have no other protection. They go and actually fly into the burning forest, mate in the flames, and deposit their eggs into the burning wood. Nothing else is going to be there to attack them or attack the eggs. Those eggs are going to be perfectly fine surviving. And then you have a burned forest. It'll take some time for anything else alive to go back there. There's nothing to eat, everything is burned, which provides protection for those eggs to hatch, become larvae, and then become beetles again. So that is infrared, but it's really heat detected. Actually, it's detecting both light outside of the spectrum that we can see and the heat. Uh, so we don't have all this kind of fancy mechanisms for photoreception in various parts of the brains and measuring different aspects of light. For us, everything is happening in the eye, and eyes are in the orbits of the skull. They're protected by bone, a big layer of fat. Uh, they're protected from dust and sweat, by eyebrows, by eyelids, by eyelashes. Uh, there is conjunctiva, that is the inner surface of the eyelids and continues over the sclera, which is the white of the eyes. There are lacrimal glands, those are tear glands, which have lacrimal ducts that put tears onto the surface of the eye, keeping them clean and lubricated. Uh, there are six extraocular muscles which move the eyes inside of the orbits. We are going to look at them in lab, and you definitely need to know them. Uh, so they originate on the bone somewhere inside of the orbit, and insert onto the surface of the eye. So there's a superior rectus, inferior rectus, medial rectus, lateral rectus. Each one of them, as it contracts, it moves the eye up, down, left, or right. And then there's inferior oblique that rotates the eye laterally, and superior oblique that rotates the eye medially. And that superior oblique has an interesting tendon that passes like a pulley around a piece of cartilage. It's called a trochlea. You'll see the picture, and you'll see it on the lab model. And then there's another muscle, levator palpebrae superioris, which is not inserted on the eye but lifts the upper eyelid. Uh, those muscles are innervated by different nerves, and but most of them by oculomotor nerve. So whole bunch of muscles moving the eye around. So the eye is a sphere, it's a bowl, it is hollow, three layers of tissue making up the eye. On the outside is the sclera, which is a fibrous connective tissue, that's the white of the eye. And there's cornea, which is the transparent portion in the front of the lens. Then the middle layer is the vascular tunic, so vascular as it has a lot of capillaries, and and in front it's called it's connective tissue that's called choroid. At the front of the eye, there is no choroid. Instead, there's ciliary body and the iris. Ciliary body is a set of small muscles uh, that are smooth muscles that are attached to the lens by suspensory ligaments. So the ciliary body bends the lens in order to focus the light, make the lens thicker or thinner, and that way focusing the light onto the back of the retina. Uh, in front of the ciliary body, in front is the colored part. That's called the iris. Iris is a smooth muscle that opens and closes the pupil, which is a hole. Pupil is a hole that allows the light to enter the eye. Now, the innermost layer of the eye is the neural tunic, which is called the retina. The retina is the business part of the eye. That's the sensory system inside of the sensory organ. A retina contains photoreceptors. That is the functional part of the eye. Uh, so, um, between the cornea and the lens in front is the anterior cavity of the eye. It contains the iris, the ciliary body, the aqueous humor, which is thin and clear liquid. The rest of the eye is the posterior cavity, which is filled with the thicker, viscous glass, like vitreous humor. So let's look at the retina, which is the business end of it, and it has several layers. The outermost layer is a pigmented layer. It reflects the light and prevents it from going any further, so it doesn't go all the way into the brain. So light hits the eye, goes through all those liquids and all that stuff, hits the retina on the back, and when it goes through all of the layers of the retina, it hits uh, the outermost layer, which is a pigmented layer. In many animals, this called is tapetum because it's reflective. When you drive around and you see two green dots or yellow dots, you see there's a deer or a skunk or something like that has a tapetum. We don't have that for us; it's just black. Uh, so kind of embedded. So if this is the pigmented layer at the back, embedded into the pigmented layer is the first layer of cells. Those are the photoreceptors. So the light is going all the way through the liquid, the whole eye, and has to go through a whole bunch of cells until it gets to the cell number one, the actual photoreceptor. There are two types of photoreceptor cells. Those are rods and cones. Rods are kind of thin and long. Cones are a little thicker. Uh, Rods and cones make synapses to the outer synaptic layer and onto the next layer, which are the bipolar cells, and then the inner synaptic layer, which synapse to the retinal ganglion cells. And then there are amacrine cells, and horizontal cells there. There is some communication here that occurs by gap junctions instead of synaptic transmission because vision has to be going on really, really fast. So some of the interaction between, let's say, amacrine and horizontal cells onto the bipolar cells might be mediated by direct signaling using gap junctions. And then, so, the light is coming from here, hitting the tapetum or hitting the pigment, and this is where the receptors are, the receptor cells, and then the next layer of cells, the bipolar cells and then the ganglion cells, and then you have the axons of ganglion cells going here and to kind of turning and all fusing for all of these parts of the retina for all of the cells uh, fusing into the optic nerve. So light has to go through a whole bunch of stuff before it actually hits the rods and cones, before it hits the photoreceptor cells, before it hits the photopigments. Uh, so the place where all these axons of all the ganglion cells from all across the retina, where they've kind of gathered together and go back into the brain as an optic nerve, that place in the retina is called the optic disc. That optic disc, where all these axons are going through, cannot have any cells, right? It cannot have any photoreceptors. So this is the blind spot uh, because there's just axons passing through. The blind spot is a little bit on the side and a little bit under. So when you're looking straight at something, you're not hitting the light is not hitting the blind spot. Uh, it's actually hitting a place which is called the fovea, which is a spot on the retina directly behind the pupil. And and all those other supporting cells, all the bipolar and ganglion cells and amacrine and horizontal cells, they're all pushed aside. And when you hit the fovea, the light goes through the eye, all those liquids, and then hits directly photoreceptor cells. And so those photoreceptor cells are exposed to light more directly. So this is the place where you have the greatest visual acuity. And it's a small number of cells, and each one of those cells connects to exactly one bipolar, which synapses onto exactly one ganglion cell. So there's a very, very great kind of fine tuning of these cells. One photon, one cell, one trigger, and all the cells in the fovea are cones, while most of the cells in the periphery of the retina are rods. So fovea is right behind, and it's the place where you have the greatest visual acuity. That's what you're using when you're reading or when you're looking at some fine detail. So photoreceptors, the rods and cones, they have pigments. They have to have colored molecules that will change, be energized when they are hit by photons. In the rods, this pigment is rhodopsin. It is used for black and white vision and night vision. So when you walk into a very dark room and you start making out the shapes of the objects, you're using only rods. They have a high sensitivity. A single photon can stimulate a rod cell. Cones have similar pigments, opsins, that are called color opsins because there are three versions: the green, the red, and the blue. And so those three different molecules are sensitive to different particular wavelengths of light. And what are wavelengths of light? They're colors. So we use those to detect colors. Uh, they're not as sensitive as rods. They need multiple photons in order to be stimulated. Uh, but they have a great ability to detect edges, shapes, and of course colors. So they're the most important part of the visual information, making the shapes, making the images that our brains can interpret, but they require bright light. You can't use them in the darkness. Both rods and cones each have two parts. There's an inner segment, there's nucleus and other organelles, and the outer segment that is specialized for photoreception. So in the rods, the outer segment is a stack of membrane-bound discs, and each disc has a rhodopsin embedded into it. In the cones, the outer segments have infoldings of the cell membrane, and those infoldings contain the color opsins. So each cone cell can only have one of the three possible options. So there's can be a red cone, a green cone, or a blue cone. You can't have them all mixed up. So we are going to be able, using those three types of cones and the rods, to see a visual spectrum from about 380 to about 740 nanometers. 380, that's kind of violet. Anything lower than that is ultraviolet. 740 is red, and everything higher than that is infrared, which is high energy light. So you go further into the infrared, it's heat. It can be detected both as light as the heat. So all the other colors that we see inside of them are that have the different wavelengths, red, orange, yellow, green, blue, and violet. They are all between 380 and 740 nanometers. So photopigments, the opsins, are transmembrane proteins. Remember transmembrane proteins? They are embedded into the membranes. Remember those discs in rods and infoldings of the membrane in cones. Uh, there is a cofactor. It's called a retinol, which is derived from vitamin A, why we need vitamin A for good vision, and the retinol is attached to the opsin, so they come together. And when a photon hits retinol, gives it energy, and that energy then gives results in a change of the shape of the molecule. So some of the double bonded carbon atoms switch from cis to trans, and this change is called photoisomerization. That's light-induced change from one isomer to another. And so, without being energized, those double bonded carbons are in cis formation. Once they are, and so it's twisted, and then and that's called 11 cis retinol. After being hit by a photon, this molecule energizes, untwists, and now it's called all trans retinol. It's a straight carbon chain. Uh, So when retinol and its opsin are energized by light, they activate the G protein, and the G protein changes the membrane potential of the cell. And so at the end of the cell, it releases neurotransmitter, in which synapses onto the bipolar cell. Occasionally, horizontal or amacrine might be there too. Uh, so the once it does that, uh, that there's a short period of time that energized photoreceptor cannot respond to light again. It's called bleaching. You hit it with another photon, it doesn't require, it doesn't respond. So there's are enzymes that revert the retinol back from 11 from all trans back to 11 cis, from the open straight to the twisted version, and then it can be activated again. Uh, so different options are sensitive to different light wavelengths. Rhodopsin for 498 nanometers. Color options are red, green, and blue wavelengths. Rhodopsin is much more sensitive. Uh, so in bright light, right, like right now, all rods are bleached all the time. All the visions by cones. In the dark, cones are not sensitive to get activated, so all the visions by rods, which are not bleached because it's dark. Melanopsin, remember those 2,000 cells, 1,000 in each eye, is also blue light pigment. Intense blue light is sometimes you can buy those blue light lamps for treating SAD, seasonal affective disorder, for treating jet lag. There are smartphones that have the setting, the evening setting that reduces the blue portion of the spectrum of the screen, so you can more easily fall asleep afterwards. So the brain is integrating the information it gets from the retina, and it compares how much information it's getting from each one of the cones, and calculates the color. And at night, rods, which is grayscale, and but our brain might insert color from memory, so you have a colored image. Although as there's a sensation is black and white, sensory perception is colored because the brain fills in the color. Uh, Horizontal and endocrine cells increase acuity and make sure that there's more of a contrast. So when photoreceptor is hit, it's these cells that are making sure that the neighboring cells get inhibited, so you have more of a contrast. So once you have all of these uh, photoreceptors talking to bipolar cells, talking to ganglion cells have all those axons going through the optic disc, forming an optic nerve. Optic nerves travel towards the brain, they cross at the optic chiasm, and after that, they're called optic tracts. Uh, Axons from the ganglion cells that are located in the medial parts of the retina, so closer to the nose, are crossing over in the optic chiasm to the other side of the brain. So from the left side, from the left eye, those that are closer to the nose, they're going to cross over to the right side of the brain, and from the close to the nose of the right eye, going to the left side of the brain. The axons that come from the ganglion cells that are from the outsides of the eyes don't cross. They go to the same side of the brain. That way, because we are binocular animals, we see in 3D because both of our eyes are in front. So this kind of a combination of crossing and not crossing allows us for, let's say, right side of the occipital part of the brain, the occipital part of the brain the uh, where the vision processing area um, is going to process the left part of the visual field, all right? And so seen both by the left eye and the right eye, inside part of the left eye, the outside part of the right eye, they're going to see the left part of the visual field. Remember, these cross, those don't cross. So it's all going to be processed on the right side of the occipital lobe of the brain. Okay, uh, the so that's how we see different parts of the visual field with both eyes. So remember that there's a small number of axons that don't go there. Uh, they they don't follow the visual pathways. They go to the superior colliculus, where the visual information is compared to the auditory, and some of the sensory information initiates the movement of head towards the source. So you saw the lion, you heard a lion, sense the sense the lion in some other way. You are going to turn your head there, right? So superior colliculus is going to put that information together. And then you have that small number of axons in the optic tracts, those 1,000 axons from each eye that come from those ganglion cells that have melanopsin, and they're going to project to three different parts of the brain: the center for mood, the center for the pupillary reflex, and to the circadian pacemaker in the suprachiasmatic nucleus on the bottom part of the hypothalamus, right on top of the optic chiasm. Uh, the visual parts are going to go, to various different parts, of course, in the thalamus, and and uh, uh, and then from there to the visual cortex and the occipital lobe. and different parts of the visual cortex recognize different aspects of vision, the shapes, the edges, the movement, the angles, and then it puts it all together. Secondary visual cortex, putting all that information, color, direction, movement, everything to make a complete picture. Uh, and then that information can then interact with it, some of the sensory and auditory information to make a full kind of understanding of the environment in which you're found. Okay, gustation or taste. It's a special sense, chemosensory, obviously. The taste receptors can sense salty, sweet, sour, bitter, umami, and fatty. Those are the tastes. The taste receptors are found on the surface of the tongue and they're embedded in papillae. Those are specialized regions of the tongue, that stratified squamous epithelium of the tongue, and then special regions of those are called papillae. There are four different types of papillae by shape. Those are foliate, filiform, fungiform, and circumvallate. Those four types are found in different areas of the tongue and they tend to have different types of receptors in them. They're not so much divided by function. Each type can sense most or all of the types of taste, and yet the taste buds uh, in the back of the tongue, for instance, are going to mostly be attuned to bitter because poisons are bitter. And so, if it's in the back of the tongue, and they can trigger gag reflex there, so you spit it out or throw it out. So inside of the papilla are taste buds, and those buds are composed of gustatory receptor cells. And it's sensitive to different types of chemicals found in the food, and they're going to act differently, depends on that. Um, so the chemical is present, it's going to bind a receptor on the membrane surface of the cell, of the sensory cell, which is going to release a neurotransmitter, and the neurons that receive that are going to take the inputs, those inputs to the central nervous system, to the brain. Uh, Salt. So when you have salt, like a kitchen salt, sodium chloride, and you dissolve it in liquid, and saliva is liquid, it's going to dissociate into sodium and chlorine ions. And so the concentration of sodium ions becomes high on the outside of the cell membrane. And that creates a strong concentration gradient. And that sodium diffuses into the cell down its own concentration gradient, and that results in depolarization of the cell, and so you get a receptor potential, and that leads to the release of the neurotransmitter, resulting in the action potential in the next cell, and next cell, and next cell, and the gustatory centers in the brain are going to detect the salty taste. Sour taste is done similarly, but instead of sodium ions, it's the hydrogen ions. So sour, what does it mean? It's acidic. What is an acid? An acid is something that in water is going to dissociate into something and a hydrogen. So hydrogen enters the cell of the uh, sensory receptor. That results in depolarization. There's a receptor potential. There's a synaptic transmission to the next neuron, and next one, and next one. And so higher concentration of hydrogen ion results in greater receptor potential, greater chance of eliciting action potential. The other sense is the sweet, the bitter, umami, and fatty requires the food molecule to bind to the surface receptor on the outside of the membrane. And so that receptor, when bound, initiates a biochemical cascade that starts with activation of the G protein, ends with depolarization of the cell, or hyperpolarization in some very special cases, very specific bitter molecules. Bitter taste is a way to detect toxic compounds. Those receptors tend to be in the back of the tongue, and the cells project to the vagus nerve, to induce the gag reflex. Uh, the taste buds that are in other parts of the brain are going to go via other nerves, facial nerves, glossopharyngeal nerves. You don't have to know that. Uh, this is all going to then go to the gustatory cortex, which is also in parietal lobe, where we then have a conscious sensory perception of taste. Olfaction or smell, also chemosensory modality. So there are all sorts of kind of smells floating around, and those smells are what? They're little molecules. Olfactory receptors are neurons. They're bipolar neurons, and they are inside of the nasal cavity, where they form what is called the olfactory epithelium. Now you remember from the lab, you handle the skull. There's ethmoid bone. Remember, ethmoid bone is building most of the nasal cavity, and that ethmoid bone has the two cribriform plates on each side of the crista galli, and there's lots of little holes going in those cribriform plates, and that's where the neurons of olfactory nerves are passing through. So the molecules that float in the air, the odorants, they get inhaled and they get stuck inside of that mucus where they bound to proteins that carry them and carry them to where? To the dendrites of the olfactory receptor cells. And that odorant protein complex, so you have the odorant, the smell from the air, and the protein that carries it, that's a complex now. It binds to the receptor on the membrane of the surface of the receptor cell. And those receptors are 7 transmembrane G protein coupled receptors, and they're going to result in a graded receptor potential. Uh, so they're going to take the information through the axons to the olfactory bulb. Olfactory bulb, actually two olfactory bulbs, are right on top of the cribriform plates. They are on the bottom of the brain, right? That's this part of the brain, roughly here, right, kind of on top of the nose, under the eyes, and that's the olfactory bulb, which you will also see in the lab on brains, on sheep brains, and on brain models. Uh, and from there, the actions go to several different parts of the brain for further processing. There's a primary olfactory cortex, also in the temporal lobe, so we can get a conscious perception of the smell. But some of the actions go to other parts of the brain, like the limbic system and hypothalamus, where they get associated with, long-term memory or emotions. Smells can elicit emotional responses, like you're smelling your childhood foods or freshly baked bread or something like that is going to have an emotional response. Or really bad smells, restroom or something like that can have an emotional response. Olfactory neurons, because they're exposed to the outside world inside of the nose, they get damaged, so they're regularly replaced. Um, each olfactory neuron has a receptor for only one possible shape of the molecule, of the smell molecule. And so the location of the receptor tells the brain what was the smell. So, hey, that cell has only a receptor for this shape. It smells like a rose or smells like a lemon. And so when it fires, the brain says, oh, the stimulus came from that cell, which is located in that place in the nose. That's the one that smells the rose or the lemon. So that's how it codes. That's how the brain encodes smell. Uh, so in order to have different shapes of molecules binding to the different kinds of receptors, we have to have a lot of receptors. The smells come in all shapes and sizes of molecules. So we have to have a lot of different uh, molecules that act as receptors for them, so they can fit like a lock and key. So we need to have a bunch of genes for those receptor molecules, and that's actually a pretty large part of our genome, as far as 16,000, 20,000 genes that we might have, a big chunk of that is devoted to olfactory receptor genes. Uh, and we are actually not such good smellers. Can you imagine in dogs how many they have? A huge portion of the dog genome is dedicated to olfactory receptors. Of course, different species are going to have different receptors depending what is ecologically and evolutionarily uh, important or relevant for them. For instance, there are some smells that we can smell and other mammals can smell. So, when you do research on smells in like, mice and rats, you use those kind of synthetic odorants. And then if you use the same synthetic odorants when you're trying to test the smell capabilities of birds, they don't respond because they don't respond to those molecules, they respond to others. So we now know that many birds have excellent sense of smell, but they are tuned into different kinds of molecules. Seagulls and albatrosses smell fish. Vultures smell carcasses. Uh, Quail have large and very developed olfactory bulbs, and it's probably using some kind of social cues. So birds can smell, although for a long, long time we thought they couldn't because we were trying to test them using the mammalian odorants. As I mentioned before, olfaction is the only special sense that goes to hypothalamus and other parts of the brain and does not have any pathways to or through the thalamus. All the other senses have centers in the thalamus. Olfaction doesn't. It goes directly to the cerebral cortex. That's because olfaction is the oldest, most primitive sense. I mean, bacteria have olfaction. And so it had a preset developmental pathway in the invertebrates and later in invertebrates that came way before there were complex brains of mammals. So we kind of inherited that kind of a pathway that bypasses thalamus. Now, another role of thalamus, another function of thalamus in the brain, is the initiation of sleep. So, olfaction, as the only sense that doesn't go through thalamus, is not involved in dreams. You cannot dream smells. You can dream, visual, auditory, gustatory, and tactile information in your dreams, but you can never dream smells. And this is why.